Good morning, everyone. Welcome to Neurology Grand Rounds at Massachusetts General Hospital. Today, our speaker is Dr. Jamie Hillis. Dr. Hillis is an assistant in neurology at Massachusetts General Hospital and instructor in neurology at Harvard Medical School. He is also the director of clinical operations for the Digital Clinical Research Organization in Mass General Brigham AI. His research focuses on validation studies of diagnostic artificial intelligence algorithms, with studies that he has led being used for 10 successful device submissions to the US Food and Drug Administration. He completed his medical school at the University of Melbourne, Australia, before undertaking a doctorate in philosophy at the University of Oxford in the United Kingdom. He completed the Brigham and Women's Hospital and Massachusetts General Hospital Neurology Residency Program and the Advanced General and Autoimmune Neurology Fellowship at Massachusetts General Hospital. Let's welcome Dr. Hillis.
Thank you very much, Mia, for the warm welcome. And thank you to the Chief Residents, to Merit and the Department of Neurology for the invitation to deliver Grand Rounds today. My talk is entitled Artificial Intelligence and Clinical Neurology. And I hope that I'll be able to convey to you some of the lessons that I've learned, and for that matter, some of the research that I have performed since I first dabbled in artificial intelligence when I was a neurology resident. I'll say since that point in time, my interest in this area has only grown. And fortunately, the technology has also grown. It's incredible that it took me less than a minute to create the image on this opening slide, courtesy of ChatGPT. Fortunately, through my position with Mass General Brigham AI, I continue to be exposed to many of the latest and greatest innovations coming to healthcare. And I'm hopeful that I'll be able to share some of those with you today. To move to my disclosures, the key disclosure that I have that's relevant to this presentation is for funding through Mass General Brigham AI for AI algorithm research, including from industry partners. As part of the talk today, there are 4 key areas that I want to touch upon. The first is to provide an overview of artificial intelligence concepts, including machine learning and artificial neural networks. The second is to detail the evolution of Mass General Brigham AI. The third is to describe some of the key lessons that I have picked up along the way about artificial intelligence implementation. And then finally, I want to illustrate some artificial intelligence use cases through 2 examples. The first is an algorithm related to intracranial hemorrhage identification, and the second is an algorithm related to ischemic core detection. So let's launch into it, an overview of artificial intelligence. And I'd like to begin with the definition of artificial intelligence. I suspect many of you have come across some sort of neurologic tool that claims to use AI. Or for that matter, you've been browsing a website that claims to use AI, or you've been part of a dinner party conversation where somebody states something uses AI. As you ask further questions, however, you realize that what they're talking about is actually something that is using exactly the same computer logic that has been used for many decades. For those of you familiar with computer programming, statements along the lines of if, then, and statements like that, and you realize that it's not what you would actually imagine for AI. And the truth is that AI has a buzz about it, and a lot of people use it as a buzzword. And at the same time, there's not a formal definition behind AI, which means that people can get away with using it as a buzzword. So here are some of the definitions from the literature. One of them is what we don't know how to do yet. The next is to make computers do the sorts of things that minds do. Now, for many of you, you probably have more of a futuristic sci-fi conceptualization of AI. And what that is actually artificial general intelligence. And this is a hypothetical computer program that can perform intellectual tasks as well as or better than a human. I like to think of this as not just an algorithm that is answering questions or completing tasks, but actually an algorithm that is asking the questions to begin with, or conceiving of the tasks. Many of you will be familiar with the Turing test. It was proposed by Alan Turing, a British mathematician and codebreaker. And this involves a human interrogator interacting with a machine and human via written statements. And in order to pass the Turing test, the human interrogator is not able to distinguish between the machine and the human. And crucially, over the past couple of years, we've seen a number of large language models pass the Turing test. Initially, Google's Lambda chatbot, now known as Gemini, passed the Turing test. Subsequently, OpenAI's ChatGPT chatbot also passed the Turing test. As you can imagine, it's a huge milestone for artificial intelligence and data science to have those models passing the Turing test. A key reason why there have been so many advances recently with artificial intelligence is to do with the increase in computing power. Gordon Moore was one of the co-founders of Intel, and in the 1960s, he proposed that the number of transistors on a microchip would double every year. He revised that prediction a decade later to every 2 years. And here in this graph, which extends from 1970 through until 2020, you can see how that prediction has stood the test of time over a 50-year period. So to go into the definitions in some more detail, here you see a Venn diagram that suggests that machine learning is a type of artificial intelligence, and then deep learning is a type of machine learning. I realize that there are some wordier definitions on the slide, but what I will say very briefly is that machine learning is about pattern recognition, and then deep learning is using neuron-inspired models to enable that pattern recognition. And there are many different types of learning that can be used. So to go through some examples, first of all, supervised learning. And this is where you provide labels so that a computer can learn to identify those labels. A classic example here is providing images to a computer model of a cat and a dog and having them labeled as containing a cat or a dog. You give hundreds of these images to the computer model, it learns how to distinguish them. The hope is that you can then give it an unseen image of a cat or a dog, and it can tell you accurately whether it has a cat. It's actually exactly the same type of process that is used when you're creating many of the different machine learning models for medicine. If you're wanting to create a model that is able to detect an intracranial hemorrhage on head CT, you can feed a model plenty of images that contain an intracranial hemorrhage and are labeled as such, and plenty that do not contain an intracranial hemorrhage, and then hope that you can use that model in the real world on scans as they are obtained. Unsupervised learning, on the other hand, is about allowing a computer to infer relationships without labels. A key example that I like here is predictive advertising online, which I suspect many of us are exposed to day in, day out. And a lot of companies use algorithms, a lot of e-commerce companies use algorithms where what they are doing is they are looking at different products that you are browsing, different products that others are browsing, and for that matter, different products that ultimately others browse and then purchase. And from this, they're aiming to learn links and relationships between different products so that they can then target you with their advertising. The key difference here from supervised learning is that unsupervised learning does not have the labels to begin with, but rather the machine learning model is learning and inferring the relationships along the way. An example in the neurologic context would be to look at discharge medications. And courtesy of the stroke service, you'd hope that a machine learning model would quickly learn a relationship between aspirin and atorvastatin, given how many people would be discharged on that combination. And then lastly here, I have reinforcement learning, which is more of a Pavlovian learning style. And this is where a computer receives and incorporates feedback. A key reason for actually putting reinforcement learning here is to emphasize that there are many more types of learning beyond just supervised and unsupervised. And I'll actually say that there are a lot of hybrid types of learning as well. And so semi-supervised learning is a type of learning that involves both supervised and unsupervised learning. And self-supervised learning is where you actually have an AI model that comes up with the labels itself. Semi-supervised learning and self-supervised learning are actually two of the key techniques that are used by large language models such as ChatGPT. And the fact that you do not need to manually curate and label that data is a key reason why it has been possible to develop such models. So to jump to deep learning and specifically artificial neural networks, as I mentioned, this involves a network that is inspired by neurons and synapses, and it's a network that goes from an input to hidden to output layers. So in explaining this, I ask you to think of a hypothetical algorithm that is aiming to detect intracranial hemorrhage on a head CT. The CT and the pixels and the voxels within the CT is the input layer. Whether or not it contains an intracranial hemorrhage is the output layer. And along the way are all of these links that have different weights that go from that input layer through the hidden layers and ultimately to the output layer. The analogy here is that the links and their weights are similar to neurons and the strength of their synapses connecting with a subsequent neuron. And when you're training this type of algorithm, what you essentially do is you actually take the expected output and compare it with the actual output, and then go back and adjust the strength and the weight of the links along the way. And by going through this training process thousands of times with thousands of head CTs, you end up with an accurate algorithm that can detect, or at least you hope that you end up with an accurate algorithm that can detect intracranial hemorrhage on head CT. For the final slide for the overview of artificial intelligence, I wanted to display a graph that shows the cumulative FDA clearance of artificial intelligence algorithms for neuroradiology. And you can see here how they really took off in 2019 to 2020. I'll say that this graph is actually now a couple of years old. It actually comes from an issue of Seminars in Neurology that Marcelo edited. But if you were to recreate this graph today, you would find that this explosion is even more profound. And so having completed an overview, I'd now like to transition to Mass General Brigham AI and explain how this group has come to evolve. And in putting together this slide, I thought I could do with some assistance. Hi, ChatGPT. Can you please create an image showing the interaction between Massachusetts General Hospital and artificial intelligence? Yes, I know it's a computer algorithm. I do still say please to it. It took ChatGPT about 20 seconds to reply. Here is the image depicting the interaction between Massachusetts General Hospital and artificial intelligence. If you need any adjustments or additional details, feel free to let me know. Now, if you zoom in on this image, you can actually see that there are a number of errors with the image. The item that stood out to me most is actually the size of the people standing outside the hospital is such that they're taller than the ground floor of the hospital. And this actually brings up one of the big challenges with large language models, which is referred to as hallucinations. It's typically actually thought of more with text-based outputs as opposed to image-based outputs. But it's when you have a large language model that very confidently gives you information that ultimately ends up being false. I will say that we could talk for hours about large language models. I do want to provide one provoking thought to you, which is that hallucinations are not necessarily a bad thing. By thinking outside the box, that then enables creativity and new thoughts, and there is certainly value there. At the same time, however, if you're relying on ChatGPT for medical advice, you want to know that you can trust it as a source. So to move back to Mass General Brigham AI and how it came into existence, in the mid-2010s, there were a number of different companies that were approaching MGH and Brigham with a request to purchase data. A lot of this was radiologic data, and a lot of these companies were ultimately hoping that they would be able to use that data to train AI algorithms. MGH and Brigham, however, indicated that the data weren't for sale, but that they were willing to partner with these companies to develop different AI algorithms. And so at that point in time, what was called the Center for Clinical Data Science came into existence. It was a team within partners at that stage, Mass General Brigham now. It was a team within partners that consisted of data scientists, software engineers, clinicians, and plenty of other people on the team. And it was dedicated to developing different AI algorithms. I first became involved at the start of 2018 and was involved with a team that was developing a suite of different stroke imaging related algorithms. Somewhat coincidentally, in the middle of 2020, as I was finishing up fellowship and becoming an attending, we were also finishing up with the development of a number of these algorithms, and we were thinking about the next steps for them. And we were thinking about how do we actually deploy them clinically? And critically along those lines, we were thinking, how do we validate them and get them through the various regulatory pathways? And while we set up the group, which we called the Digital Clinical Research Organization, while we set it up for our own internal algorithms, we actually found that there was a huge interest from companies for validating their own algorithms before they took them to the FDA. And actually a huge interest from companies outside the US, because the FDA typically will want a company to have US data, as in US radiology studies, that algorithm has been assessed on. And they will also want involvement of US board certified radiologists or other clinicians. And fortunately, we were able to help out with the US data. We were able to install the models within Mass General Brigham and then run the models within the Mass General Brigham firewall. We were also able to help out with US radiologists and other clinicians. And for that matter, we were able to put together a team that was able to take care of the study design and execution. There were 3 of us initially, Bernardo Bizzo, John Chin, and myself. We've now grown to a team of roughly 15, many of whom are here today, whether it's in person or virtual. And just to formalize what I went through on the prior slide, the initial life cycle of AI algorithms involves ideation and fundamental research, early development, late development, and then regulatory approval and clinical deployment. And within Mass General Brigham AI, at least over the past couple of years, we've had 2 different teams. One is the product development team, and the other is the Digital Clinical Research Organization that work on different parts of this process. Some of the items that we're involved with include product design and market fit, data curation and annotation, AI product development, validation strategy, product performance assessment, and then deployment and monitoring. There are two notes that I want to make of other things that we've done along the way. And one takes into consideration that ultimately we are producing software as medical devices with the hope that they will be able to go through regulatory processes. And so as part of this, we have needed to have very defined processes and documentation. And so we have developed and maintained a quality management system to enable us to have those processes. In addition, we've developed a number of other tools. One of the most critical tools is something that we refer to as the AI control panel. This is an annotation tool that radiologists and others are able to use and go through the different images to say present, absent, or whatever else they may be looking at as far as what they see on a radiology study or some other type of image. And so the digital CRO was created in 2020. I'll say that this image is actually a few years old now, and it only contains a subset of the team members who I'm lucky enough to work with. But we have now worked with 12 different companies for both regulatory and non-regulatory purposes. And these projects have encompassed approximately 50 findings and 6 modalities, including X-ray, CT, MRI, point-of-care ultrasound, ECG, and vital sign data. And when I think about what we've accomplished over the past 4 years, the thing that I actually think about most is that our projects have now been used as the basis of 10 successful device submissions to the US Food and Drug Administration. I still remember how exciting it was when the first device was cleared by the FDA towards the end of 2020, start of 2021. And it's incredible to think that we're now in double digits. I'll actually say that number 11 is not that far away. The FDA process works very similar to submitting a manuscript to a journal. The FDA had come back for revisions, and hopefully those will go back to the FDA actually today or tomorrow. And then hopefully over the next month, we'll hear some positive news. Devices number 12 and 13, we've actually already finished the studies for. Hopefully those will be going to the FDA soon. And devices number 14 and 15, we're in the process of completing those studies at the moment. So we're definitely hard at work. As far as lessons about AI implementation, What I want to capture here are some of the key facts and key lessons that I've learned along the way, ultimately through the exposure to many different parts of this regulatory process. And the first is that implementation of AI will occur via incremental steps. So here you can see some proposed steps where AI can help the radiology workflow. Specifically in blue, you can see different steps of the radiology pathway. You've got a clinician, they submit an image request that leads to acquisition data. It leads to DICOM images, that's the file format of radiology images. That then leads to a radiologist interpreting the DICOM images. Radiologist produces a radiologic report. It leads to a diagnosis and ultimately health outcomes. In addition, on this slide, you can see a number of the proposed ways in which AI can help. And this is in the dark text. And so, for example, in the middle at the top, you can see protocol automation. This is where you use an AI algorithm, for example, with an MRI to determine which sequences you will require on that MRI. Or, for example, whether or not that MRI will require contrast. Similarly, if you just move along a little bit, there is image enhancement. And there are plenty of opportunities here, but one of them is actually you can potentially use a lower dose of contrast with MRI and then use an AI algorithm to bring out that contrast more on the images. To segue to what I will talk about in my next slide, I want you now just to focus on the three words that are in the top right of that image, which are detection, segmentation, and classification. When it comes to an AI algorithm interpreting an image, there are a number of specific tasks that it can perform. Detection is, as the name suggests, it involves detecting whether or not a finding is present. So in the case of an intracranial hemorrhage, it would be whether or not the intracranial hemorrhage is present or absent. Segmentation is to do with segmenting, drawing a line, a circle, etc., around the object and determining what the region of interest is. Classification is providing some sort of further discrete output. So in the example of an intracranial hemorrhage algorithm, it would be specifying whether that hemorrhage is an epidural, subdural, subarach, so on and so forth type of hemorrhage. And so the reason that I bring this up is that it ultimately relates back to what is allowed by regulatory bodies who think very much around the task of what AI algorithms or what the task that an AI algorithm is performing. And so lesson number 2 is that regulatory body approval provides a mechanism for balancing the potential risk and benefit of devices. And in the US context, the FDA process, especially the 510(k) pathway, has led to lanes for different device types. To provide some background here, AI devices are typically classified as Class 2 devices by the FDA, and there are 2 broad pathways for devices working their way through the FDA, or for Class 2 devices working their way through the FDA. One is the de novo pathway. The second is the 510(k) pathway. As the name de novo suggests, this is where it is a new type of device for the FDA, and the FDA needs to consider the risks and benefits of such a device. And ultimately, the FDA will create what is called a regulation for this device, and specifically special controls that specify what you can and cannot do for that device. The 510(k) pathway, on the other hand, relies on what are referred to as predicate devices. And in that sense, they actually rely on the risk assessment and the special controls of devices that have come before them. And so here is a summary of some of the key radiologic computer-aided device types. I'm only going to go through 3 of them. The first is computer-aided detection. And this is the concurrent or sequential use to aid the identification of potential disease. An example that I like to use here is for an MRI, you could use a computer-aided detection device to show you T2 flare hyperintensities. Critically though, what detection does is it shows what is there radiographically. It does not actually take the next step to interpret that and provide you with a diagnosis. And that's where computer-aided diagnosis algorithms come in. Here, you would actually have that MRI algorithm, but have it actually be able to tell you whether or not the T2 flare hyperintensities that you're seeing reflect multiple sclerosis or NMO or ADEM or another condition. I will note that the text on the right of this slide suggests that CADX algorithms are specific for cancer. Actually, through the next item here, which is the combination of CAD-E and CADX devices, that is expanded to many other conditions, including neurologic conditions. And the third type of device that I want to go through are computer-aided triage and notification devices. And so this is where you end up with notification of a potentially time-sensitive finding. Many of you will be familiar with the fact that MGH has used the VIS large vessel occlusion device on the stroke service and through the telestroke network to identify large vessel occlusions on CTA scans that are being performed. The VIS device was actually the first computer-aided triage device to come through. And ultimately, the purpose of such a device is when you've got a radiologist, or for that matter, a neurologist who has 100 different studies that they're aiming to work through, and the abnormal one is number 99, these devices will actually promote that study to the top so that it can be interpreted first rather than interpreted last. And just to jump back to the 510K pathway briefly, I'll say VIS received or were granted their de novo request at the start of 2018. To give you an idea of how many devices come through on the 510K pathway, there have actually been 83 devices that have followed on as CAD-T devices since that VIS device came through. Lesson 3 is that FDA lanes can lead to manufacturers curtailing innovative features that extend beyond a predicate device. And here you can see the outputs from the same pleural effusion detection algorithm as the CE mark device in the European Union, which is on the left, and the US FDA cleared triage device on the right. So as far as this pleural effusion is concerned, on the left, you can see what we refer to as a heat map, which is where you can see the different colors suggesting that that is what has caused the algorithm to detect a pleural effusion that is present. In the sense that the CAD-T triage devices are there to prioritize the work list, literally all you can do with the same algorithm that has been cleared in the US is have the pink dot come up that tells you that a pleural effusion is present. In case you're wondering, the blue dot there indicates that there is a pneumothorax, a different finding on some of the other scans. But it's something where the fact that the FDA does have these lanes, it actually leads to many manufacturers curtailing innovative features that extend beyond a predicate device. And that's ultimately because the first devices that came through had these special controls, and the subsequent devices need to think about the same special controls. I will say I find this a personally conflicting area because I very much appreciate the FDA needing to consider risk mitigation and ultimately ensure safety. But at the same time, by lumping everything in together and not considering each algorithm and performing a holistic assessment of each algorithm, they don't get the opportunity to use some of the features that could actually be clinically beneficial. And I'll say the key example actually of where CAD-T triage devices could benefit from what we call explainability, and that is having something similar to a heat map that shows what has caused it to be positive, is actually for false positive studies. So this is where the algorithm falsely identifies something, and then you as the clinician come along, you're saying, where is the pleural effusion? Or for that matter, with the VIS large vessel occlusion model, where is this large vessel occlusion that I'm meant to be detecting? And at this stage, because of the FDA limitations, you don't have that explainability to say what's going on. Lesson number 4 is a counterpoint to the prior slide. And this is that the FDA still provides plenty of opportunity to innovate. So here is a device that was granted a de novo request by the FDA earlier this year, and it established a new regulation for devices that predict Alzheimer's disease. Specifically, this device is called BrainC, and it provides a similarity score to predict amnestic mild cognitive impairment progressing to clinical AD dementia within 5 years. Now, there were a number of key things that stood out to me about this algorithm. The first is that it's a prognostic algorithm. And so with many of the other algorithms that I've mentioned, they are diagnostic. And ultimately, you can verify those findings in real time for whether or not they're present on the chest X-ray or on the CTA. When you're dealing with a prognostic algorithm, as many of you all know, the only way you really confirm whether somebody has Alzheimer's disease is to see how their clinical course evolves. And so when people talk about the black box of AI, here, the AI really is a black box, and it will take time to actually see what happens with each individual patient to see whether or not the AI has been accurate in predicting whether or not they'll develop Alzheimer's disease. Secondly, I'll say this particular algorithm was multimodal. It actually incorporates patient demographic data, their age, their sex. It incorporates clinical dementia rating scales, including the MMSE. It also incorporates interpretation of the patient's MRI. And having multimodal algorithms, I'll say, is very much one of the key next steps for AI algorithms, moving beyond simply interpreting a single CT or MRI or chest X-ray. The third item that struck me about this, and I'll say this is actually more thinking about the clinical consequences moving forward, was thinking through how will this actually be used? And especially in the world where we have amyloid monoclonal antibodies, how will this be applied to clinical care? And you can very easily see some sort of algorithm along these lines, A, being used by clinicians to say whether or not a patient does indeed have Alzheimer's disease and whether they should receive lecanemab or another monoclonal antibody. B, it could actually be used by clinical trialists to select patients for clinical trials. And then C, it could actually be used by insurers and payers to determine whether or not they will actually cover lecanemab and other similar medications for patients. And I don't think that area has been thoroughly thought out yet, but it will be something that will need to be thought about over the months and years to come. Finally, here, in terms of the lessons, we are only at the tip of the iceberg. It is easiest to conceptualize how AI can replicate human tasks, but the much more exciting bit is how AI can augment neurology and medicine more broadly through beyond human tasks. And now I'm going to launch into a couple of case examples. These actually feature algorithms that we have very much been involved with. The first is an algorithm that we had performed a validation study on ahead of it being submitted to the FDA. The second is actually a homegrown algorithm that we had worked on. I'll actually say that Anish was part of one of the studies involved with that algorithm. So with the first algorithm, it's an intracranial hemorrhage identification algorithm. As I was explaining for CAD-T triage devices, the automatic identification of intracranial hemorrhage on non-contrast head CT would allow radiologists to prioritize or triage interpreting cases with suspected hemorrhage. Once again, if they've got a list of 100 cases they need to interpret and the abnormal one is number 99, they can interpret it sooner and have the patient on the right track of care sooner. The Annalise algorithm, once again, which was an algorithm that we had validated, was the first FDA-cleared CAD-T triage device to separate out the identification of 4 hemorrhage types: acute subdural and epidural hematoma, acute subarachnoid hemorrhage, intraaxial hemorrhage, and intraventricular hemorrhage. We referred to this particular type of study as a standalone model performance assessment. This is where we're assessing how well the model performs, its sensitivity, its specificity, etc., for identifying a hemorrhage. And so here we selected 4 different cohorts for the 4 different hemorrhage types that consisted of over 2,000 cases in total. We then completed ground truth radiologist consensus interpretations. This involves sending out studies to 2 radiologists initially. If they both say it's positive or both say it's negative, we've got a consensus interpretation. If they're split, then we end up sending it to a 3rd radiologist for them to provide an adjudicating view. And then we perform model inference on the thin and the thick series from these studies. And so we actually ended up with, courtesy of having the thin and the thick series, and then the 4 different cohorts, we ended up essentially with 8 different sets of results for this particular study. And so here is one of the results. It's for acute subdural and epidural hematoma on thick series. And its performance at an operating point, I will explain what operating point means in a moment, but its performance at an operating point of 0.060177 was such that it had a sensitivity of 82.5%, a specificity of 89.9%, and an area under the curve of 0.942. Now, to delve into a few further specifics about how these models work, this is a classification model. This is a detection model. It is detecting whether or not a CT has an intracranial hemorrhage, and specifically an acute subdural and epidural hematoma. And as part of determining this detection, it provides what we refer to as a classification score or a confidence score. And this is the number that you can see in the top left of each of the images. This is a score between 0 and 1 that indicates the confidence of the model that there is, in this case, an acute subdural or epidural hematoma present on the scan. And so you can see with a very obvious subdural hematoma, the model gave it a score of 0.98. In the second one, there is a more subtle hematoma that's present along the midline. The model gave it a score of 0.72. And in this one, it's even more subtle. It's here in the anterior folks, and the model gave it a score of 0.23. Here is one where the subdural hematoma was adjacent to the posterior folks, and the model gave it a score of 0.03. This one, due to the threshold of the model, actually ended up being a false negative, and so the model would have missed it. So I'm just going to jump back to the operating point here and explain what this means. And this is the threshold for calling a case positive. And so ultimately, if this number comes back greater than, in this case, a 6 decimal place number, 0.060177, it would be considered positive from the model. And similarly, if it comes back below that number, it would be considered negative from the model. And so that's why this one here is considered a false negative case for the model, and these are all considered true positive. This case here was actually a false positive case that the model identified. I'll say that there's no agreed upon approach for how to determine operating points and thresholds. Many of the companies that come to us have their own individual approach. Actually, many of them don't even release to the public what their operating point is. In this case, this is actually a publicly known operating point. But the thresholds are ultimately determined for each individual algorithm and determined such that appropriate cases are being called positive, appropriate cases are being called negative. And that also considers what you want the positive predictive value and the negative predictive value for an algorithm to be. Here is a graph that we refer to as a receiver operating characteristic curve. This is based off the operating points, and this essentially shows what the sensitivity and specificity would be if you chose different thresholds along the way. And so in the top right corner here, you can notice that the sensitivity is 100%, the specificity is 0%. That would occur if you used an operating point of 0 and called everything positive. And then conversely, if you look at the bottom left corner of the graph, you have a sensitivity of 0%, a specificity of 100%. That would be where you have an operating point of 1. And of course, you've got all of the points in between. And this particular point here corresponds to the operating point in the slide. So with submitting these sorts of analyses to the FDA, these standalone model performance assessments involve extensive subgroup analyses to ensure generalizable performance. Here you can see the subgroup analyses for sex, age, ethnicity, race, and manufacturer. And for this particular program, this analysis was repeated for the 4 hemorrhage types and each of the thin and thick series, and it was ultimately repeated 8 times. I'll say when we need to work through all of these subgroup analyses, which are ultimately exceptionally important, but when we need to work through them all, we end up with some quite lengthy study reports. Sarah McCaldo, who is the biostatistician on our team, and I, we frequently joke that these reports end up being longer than our PhD theses. Actually, for many of them, we end up with reports that are longer than our PhD theses combined. There's a 409-page document that's on my to-do list for the days ahead. In thinking forward about how this algorithm will be used and what are the next steps for it, the overall Annalise Enterprise CTB algorithm can detect 130 findings on non-contrast head CT. I'll say that not all of them are currently available in the US. But in the age where we have large language models that are able to generate text, you can imagine that you can use the output of these findings to then generate a draft radiology report and ultimately help radiologists with their workflow and writing their reports. And of course, that's something that Annalise has thought about. This was an announcement that was made at the time of the RSNA meeting last year, with Annalise saying that they were moving towards developing comprehensive draft radiology reports. I'll say that the same is very much in scope for neurology. It's actually something where Nuance has developed an AI algorithm that can essentially listen in on clinic visits and then provide you with a clinic note that is drafted for you at the end of it. From my end, I will say that I'm very excited about using large language models to generate prior authorization letters and letters of medical necessity. In addition to this, there has been a large language model that has been trialed within Epic at MGB to draft responses to patient messages. I'll say that at this stage, it's been something that has not been able to be used for clinical comments back to patients, but hopefully that will be a step that we're able to move forward with at some stage soon. To move to the second algorithm, which is about ischemic core detection on non-contrast head CT, I will say that this is very much for me an algorithm that encapsulates the below the water in the iceberg as something that reflects more of a beyond human task. And so here, what we were considering is detecting ischemic core on non-contrast head CT. And as many of you know, CT is quicker, lower cost, and more widely available for acute stroke imaging. But on the other hand, MRI is better at detecting acute infarct. We were fortunate to have a data set from MGH and Brigham that incorporated patients who had paired studies such that they had a CT and an MRI that were obtained within 3 hours. At least that was our cutoff for the positive pairs. And then a number of cases that were within, or a number of pairs that were within 5 days, and that was for the negative pairs. What we did was we took these CTs and these MRI studies and we registered them together so that they were spatially aligned. We then segmented the region of diffusion restriction on the MRI such that we had a segmented tracing of the region of diffusion restriction or the ischemic core. And then what we did was we took those tracings, and because we had registered the CT on the MRI, we were able to take those tracings and put them over the CT, such that we then had a set of studies where we had the CTs and the tracings over the ischemic core, and we were able to feed the CTs and the tracings into an AI algorithm. The hope was that the AI algorithm would be able to learn how to detect early signs of acute ischemia and to ultimately enable us to see some of the very, very subtle signs of early acute ischemic stroke. I'll acknowledge that Roman Garial was the data science lead for this particular project, and Bernardo Bizzo was the clinical lead for this particular project. So here you can see some examples of studies that the model interpreted. Here on the left is the CT with the model output in green. In the next column is the original CT scan. You can see that the signs of acute stroke are very, very subtle. In the next column is the DWI, and then in the final column is the ADC sequence that were taken from the paired MRI. And so not only was it incredible that the model was able to pick up this acute ischemic stroke, it's actually incredible how accurate it was with its region of interest segmentation and also with its volume estimate for the size of this ischemic core. On the next slide, I'll present a further study that we did where we actually asked 3 very experienced neuroradiologists to look through a series of CTs to say whether or not they thought acute ischemic stroke was present. These two studies, these two cases were actually part of that study. And I'll say that all three of those neuroradiologists called these two head CTs negative for acute ischemic stroke. And so this is the study that I was just referring to. It involved the comparison of model performance with experienced neuroradiologists in detecting ischemia on CT head cases. And so we had 90 CT studies that had a positive MRI pair, 60 that had a negative MRI pair. You can see the model output or the model results here in blue. The reason that it's a line and it's a receiver operating characteristic curve is that we use different thresholds of what the model was detecting to ultimately call a case positive. And ultimately, we hypothesized that it would be best to only call a case positive if it had an ischemic core of at least 5 mils. And that was to avoid small regions of noise on the head CT falsely triggering the algorithm. And so this star here corresponds to a threshold of at least 5 mils. The three neuroradiologists are reflected here with the yellow, green, and red dots. And the shading that you can see around the line and around the dots reflects the 95% confidence intervals. And you can see that there is no overlap between the 95% confidence intervals for the model and the experienced neuroradiologists. And so here, the model sensitivity was 78%, the model specificity was 98%. And for radiologists, their average sensitivity was 64%, their average specificity was 91%. This model was also able to generate the volume of the ischemic core. And so here along the y-axis, you can see the volume from the model output. And here along the x-axis, you can see the volume of the ground truth based on the MRI. And you can see how accurate the model was as far as determining the volume. The correlation coefficient here was actually greater than 0.98. I will also add that there is some color coding here to show whether or not the neuroradiologists had correctly identified whether there was an ischemic core present. Green reflects that all three of them said that it was present. Blue reflects that one or two of them said that it was present. And pink reflects that none of them said that it was present. And this particular dot here reflects the first case on the prior slide. So moving on from that, we actually wanted to see how clinical readers performed when they were aiming to detect ischemia with or without the model output. And we refer to this particular type of study as a clinical reader performance assessment. And here the question is not how is the model performing as far as sensitivity and specificity, but rather how do the clinical readers perform when they're using the model versus how do they perform when they are not using the model? So we had 8 clinical readers, 2 emergency providers, 2 neurologists, including Anish, 2 emergency radiologists, 2 neuroradiologists, and they each interpreted 180 cases, both with and without the model output. They interpreted half of the cases with and without the model as part of the first session, and then they split the cases around and interpreted them the other way as part of a second session that was 4 weeks after the first session. Here you can see 2 example cases. On the top row, it's the same case. It has the model output on the left. It does not have the model output on the right. And similarly, there's a second case here. I realize that it's quite small as far as being here on the slide, but here the volume of the ischemic core was also provided to the clinical readers. And so the clinical readers were asked, first of all, does this case contain ischemic core? Then they were also asked about their confidence of whether or not there was an ischemic core, and they were asked about the volume of the ischemic core. And these were the results based on whether or not they felt there was an ischemic core. And similar to using a confidence or classification score for a model, we were able to use the confidence score from the readers to generate receiver operating characteristic curves. And here you can see that their area under the curve increased from 0.696 to 0.836. Their sensitivity increased from 53.9% to 66.9%. And their specificity increased from 78.9% to 89.2%. And all 3 of these differences were statistically significant. In addition to that, we had asked the readers to estimate the volume of the ischemic core. And here you can see 2 confusion matrices that demonstrate the results. At the top, we have a confusion matrix that was with model output. At the bottom, there is one that is without model output. And ultimately, what you want to see with a confusion matrix like this is that the cases fall in along the diagonal, because that indicates that the physician volume matches with the ground truth volume. And so actually, if we look at the bottom right corner here, you can see that the agreement when using the model for strokes or ischemic core that was at least 50 mils was 73.8% when using the model, yet it was only 27.1% when not using the model. And if you jump back one cell, you can see that for ischemic core between 20 and 50 mils, it was 58.8% when using the model, yet it was only 30.4% when not using the model. And actually, the majority of the time, the different clinical readers were actually underestimating the size of the ischemic core that they were looking at. I will say that if you were to jump back one further box here, you will see that without model output did perform better than with model output. This is for 0 to 20 mil ischemic core. And ultimately, the clinical readers, similar to what I was describing before, were only shown the model output if the model identified something that was greater than 5 mils. So there are actually many cases with very, very small ischemic cores that were not shown to the clinical readers, and it was not a huge surprise that we didn't see the same benefit for this particular group. So that brings me to the end of my talk today. I wanted to recap briefly and go through 3 parting thoughts about how I see AI impacting neurology. I realize the first 2 here repeat a couple of the lessons that I'd mentioned earlier. But the first is just to reiterate that we're at the tip of the iceberg at the moment. It is easiest to conceptualize how AI can replicate human tasks, but the more exciting part is how AI can augment neurology through beyond human tasks. The second item is that Rome wasn't built in a day. AI will gradually and incrementally be introduced for specific tasks, for example, triaging abnormal radiology cases, rather than arriving overnight. And then finally, I think many of us have heard over the years how medicine is an art and not a science. Neurology will involve more consideration of probabilities, but a key role for healthcare providers will be the interpretation of these probabilities, and together with the probabilities, the uncertainties of those probabilities for patients. Thank you very much. Thank you as well for everybody who has made all of this work happen.
And a lot of this is tied to the industry. It's really collaboration.
Thank you very much, Merritt, for the question and the comment. Just to repeat the question for those of you who are watching virtually, Merritt was asking about ultimately, a lot of this is performed in collaboration with industry sponsors, and ultimately, what is the balance between industry and the broader academic side? And I'll say, ultimately, it's a constant case of juggling and balancing. It's something where, at the end of the day, the industry sponsors ultimately enable a lot of this research. I'll say the flip side here is that ultimately the key purpose for a lot of companies is getting through the FDA process, and that doesn't always align with the more academic, interesting side of things. Having said that, the FDA process is only the first step for actually getting an algorithm out into the clinical environment. And ultimately, there are many more steps along the lines of, well, how does it actually help patients in the real world once it's got through FDA approval? And it's actually something that we're seeing more and more now as a team. We've got plenty of companies that have got through their regulatory approval, but they're now actually thinking about, okay, how do we really prove the benefit and the use of this algorithm? And I'll say that that brings up many, many further questions. It's something where I often joke that, and I've lost track of just how many actually intracranial hemorrhage algorithms, but also lung nodule algorithms that we have worked on as a team. But it's something where at the end of the day, sort of, yes, we need to repeat these to different sponsors who are asking for very similar things. But ultimately, we're now getting into the crux of, okay, how does this really help clinically?
James, that was great. Thank you. Let's talk briefly about risks. And by that, I mean, thinking about the radiologists that are now getting presented with AI-driven measurement of stroke, hemorrhage, or whatever. What's the risk that they miss other things? Gravitate to what they presented, and the previous process of detecting all the images and thinking broadly about which, yes, interpreted influenced by what the requisition says, that question mark is obviously focused on the camera. But here they're even more focused. What's the risk?
It's a fantastic question, Andy, and I'll say there's a lot of discussion within the radiology community about how do you best use this sort of algorithm, as in, do you turn it on at the start? Is it something where you actually interpret the case and then turn it on after that? I'll say it's one of the reasons why, and I didn't delve into all of the reasons, but it's one of the reasons why the FDA has not allowed segmentation or the region of interest to be displayed to clinicians. And that's to do with the fact that the FDA, when you're displaying that sort of item to radiologists or other clinicians, they want to do a clinical reader performance assessment to actually see how the radiologists or others are impacted by having that finding shown to them. It's something I completely agree with you, there's risk. I will say though, I don't think it's actually that much different to many of the risks that already exist. As you said, sort of when somebody orders a study, there'll be a comment that actually highlights, okay, this is what the ordering provider is interested in. Similarly, actually, it's something where, for at least in an academic institution, many of the studies will be initially interpreted by a resident or a fellow. And there's the possibility that the radiologist who's then interpreting the study, the attending radiologist after the resident or the fellow, may have some sort of bias based on what the resident or the fellow has said. And so I think it will be something that's very similar to that sort of logic that needs to go into, okay, well, how do I actually start from scratch so that I make sure that I'm not actually biased as I'm interpreting this?
James, congratulations on the beautiful work and on the new fatherhood. We're hoping to see the pictures of baby William Hillis. Can you imagine prepare the talk while we're having to care for the baby?
I think my son is watching at the moment, so I will say hi to my son and also my wife Cassandra, who's looking after him at the moment.
You gave us a terrific roadmap on augmenting or improving detection or informing the technology of what we know. Can you speak also to your observation in clinical neurology and how AI will lead to discoveries of what we don't know? You have a question specifically in brain health. We hear this buzzword, but we don't know what's the measurements or really what's the longevity of the brain, how AI can inform this with the large language models.
Fantastic question, Marcelo. And just to repeat the question for those who are listening in, it's how will AI really help out with neurology? And I would come back to the prognostic algorithm that the FDA granted the de novo request for earlier this year that's providing a prognosis for Alzheimer's disease, and say that I think one of the biggest challenges that we have as clinicians is interpreting the vast quantities of data that are presented to us. And beyond that, it's also drawing the connections and the links between the different parts of those data. And so I think for many of us, if we're seeing somebody with a possibility of Alzheimer's disease, it's something where we will look at the temporal lobes on the MRI, But at the end of the day, it's not a perfect science of looking at the MRI and interpreting that. And there's the possibility that there are all sorts of other factors that are involved there, including how that pattern in the temporal lobes correlates to the person's age, their gender, and for that matter, the different clinical dementia scales that are used. I'll say beyond that, one of the use cases that I think about a lot is actually for MS, where when somebody comes to you with a new diagnosis of MS, you're actually thinking of which medication should I potentially start this person on? And it's possible that AI, through being able to learn from thousands of MRIs that have been acquired for patients, AI will actually be able to say, okay, I can predict for this person that they're going to have a more aggressive form of MS, and therefore you may consider a stronger disease-modifying medication to treat that person with. And that's something where there's sort of a beyond human insight that AI could really deliver.
Big picture question, let's say I present AI so it doesn't absolutely and then we support. Think about this, to get trained, to sign off on this.
It's a fantastic point. And I'll say from the radiologic perspective, one of the biggest challenges separate to AI is the fact that the volume of radiology studies continues to increase and increase. And yet there is a workforce shortage of radiologists. And so one of the big use cases that has been proposed is how much do you actually need a radiologist to interpret every single CT scan, MRI, chest X-ray? And so it's exactly the same along those lines. Whether or not you actually end up with a neurologist who's signing off on it, or for that matter, it's just the AI algorithm that's interpreting it. It's a big use case, and ultimately it's something that's referred to as detecting whether or not a study is normal or abnormal. And if it's abnormal, then it would go to a radiologist for interpretation.